The wandering desert spider is a species of huntsman spider that occupies one of the harshest and driest places on the planet, the dunes of the Namib Desert. The dunes are an ever-shifting sea of iron-red sand, where temperatures are highly variable every day, month, or season, blistering heat during the day and freezing during the night. How could anything live in such a barren and lifeless environment? Cue the wandering desert spider, otherwise known as Leucotrestrus arenicola, a species of burrowing huntsman spider that has made its home in the dunes of the Namib. It is a creamy white colour with jet black spinnerets and an astounding ability to learn. Dr. Thomas Norgard and a team of scientists have conducted several studies on this species of spider and determined several factors. The males of this species perform nocturnal walks when mate searching. These males can make a beeline straight back to their burrow after meandering around for a mate. They have observed a learning behaviour typically exhibited by bees when leaving the nest or burrow for the first time. This investigation will look at the plasticity of the wandering desert spider's brain. There is currently no literature on huntsman spider brain plasticity in any species. We will look at how and if the brain changes when the wandering desert spider is placed in different environmental conditions. If a change in the brain is observed, this could be an indicator that other species of huntsman spider have also evolved with the ability to adapt their brain in certain ecological stresses. This investigation is important in furthering our knowledge on the evolution of brain plasticity and which environmental triggers may have caused this trait. The aim of this research is to determine if the brain of male desert wandering spiders has a plastic response to a change in female population density. In the invertebrate brain, the mushroom bodies are responsible for memory storage. We hypothesize that when a male wandering desert spider develops in a low female population density, he will have larger mushroom bodies. Having to travel further to find a mate, the spider will need to remember how to travel back to his burrow. We expect to see the opposite in males raised in high female population densities. So how will we do this? Our study plans to have a sample size of 40 male spiders split into two groups. 20 will be tested in low female population density pens, and the other 20 will be tested in high female population density pens. The pens will be 20 metres by 20 metres based off how far these spiders will wander searching for a mate. They will be located in an arid and sandy area to imitate their natural habitat. Each set of spiders will be tested over a 2-year period to allow development and maturation. Every week, each individual will be caught, rested on ice until lethargic, and then scanned using a microcomputed X-ray tomography, otherwise known as a very small CT scan. We will examine the brain and take measurements on its size. The spiders will then be released back into their enclosure. Spiders are animals that have successfully distributed around the globe. They inhabit many ecological niches and can still thrive in human-impacted environments, and yet not much is known about them. This study will benefit the growing knowledge on spiders around the world. We do not know how plastic the brain of a huntsman spider is, nor the evolutionary function of brain plasticity in huntsman spiders, or what may trigger brain plasticity. Thus, this investigation will be the first to shed light on important evolutionary questions, such as why did brain plasticity evolve? There is also another perspective on the importance of this research, and that is how can this study benefit humans? Even after hundreds of years of medical science, we are yet to understand our own brains entirely. The outcomes of this study may unlock areas of the brain not yet considered or even studied.